We've had a little bit of practice with terminology and vocabulary, so try to unscramble some of these words. Just think about it for a sec and then we'll go over them. So what is the first one?
Cell.
Cell. The second one?
Nucleus.
Nucleus. The third one?
Membrane.
Membrane.
Membrane. Good. The fourth one? That's a little bit harder. That's a tough one. I know. It's really long. It's phospholipids. That's part of the cell membrane. 5?
Is it animal?
Animal, yeah. So animal cells, which are different from plant cells. So that's 6. Did anyone get 7? That's also a difficult one if you haven't taken the first year biology. So it's a protist. Have you heard of protists?
No.
Before? No, so protists are largely overlooked, and you know what they are. You've heard of them before, but you probably didn't know they were protists. So we often think of diseases as being caused by just merely bacteria or viruses, but there are lots of diseases that are caused by single-celled or unicellular organisms called protists. So malaria, for example, is caused by a plasmodium that's transmitted in a mosquito. It's neither a bacteria nor a virus, it's a protist. So protists are eukaryotes. They are different from bacteria, which are considered prokaryotes. So pro means before, karyote means nucleus, more or less. And so prokaryotes do not have a nucleus. Bacteria do not have a nucleus, but eukaryotes do, and that's what distinguishes us from them. What about 8? Did anyone get that one? That's another tough one. That is bacteria. A lysosome. So those are inside a cell. We'll look at those. And ribosomes. We'll look at those structures. So this is about cellular structure, the cell surface, membrane transport, the cytoplasm, and organelle function. We'll look at all those things. But how did people know in the first place that life was made of cells? It was the development of a theory called cell theory. It took quite a long time. It took quite a few people that were looking into cells and small things, but especially it took the invention and the improvement of microscopes. So Robert Hooke, he used microscopes, he invented microscopes, he named the cell. He called it a cell. So he was one of the investigators into the cells, into small things that lived, for example, in people's teeth. Schwann, of Schleiden and Schwann, stated that all animals are made of cells. That's cell theory. We now know that all life is made of cells. Pasteur, he disproved the idea of spontaneous generation. That means that living things arise from non-living matter. So people thought that for a very long time. And the Egyptians, for example, they noticed that frogs would come out of the mud of the Nile River. And they thought that frogs were spontaneously generated from the mud. But it turns out we know now that cells only come from other cells. There's no spontaneous generation. So modern cell theory emerged. So this is Robert Hooke, and he looked at the cork of plants, and he saw these little cells. They're empty cells, but they're cells, and he thought that they looked like the cells that monks lived in monasteries. So that's where the name came from, cell. Of course, they made loads of fantastic drawings using the microscopes at the time. Wrote a book called Micrographia, and we've always used that term. When you draw things from a specimen under a microscope, it's called a micrograph. Anton van Leeuwenhoek, he was also an investigator under things you could see in the microscope. So he would take small pond samples and look at that. And he called them animalcules. So this is a sample that he would have taken from a pond. And interestingly, it includes some unicellular organisms, but some multicellular organisms like this one, which happens to be a nematode, are smaller than unicellular ones. So there are some single cells, protists, they live in water and they're quite large, even though they're just one cell. And amoeba is one of them. So there is a type of amoeba that can cause dysentery, amoebic dysentery. Yeah. Cells come in many, many different shapes. And there are functional reasons for those shapes. So just like any other structure that we thought of before, the shapes of the cell fit their function. They always fit their function. So we have, for example, squamous cells. These are squamous cells. They are flat and there are single layers of squamous cells and there are stacked layers of squamous cells. Polygonal cells have a very hexagonal shape and they pack very tightly together. Cuboidal cells are like cubes, and there is quite a lot of cytoplasm relative to the surface area of the cell in cuboidal cells. Columnar cells, on the other hand, the ones that line your intestines, they're long and skinny and they have a lot of surface area relative to their volume. And that's because the columnar cells function in absorption. So they have a relatively, columnar cells, let's just write that down here, a large surface area to volume ratio. An increased surface area means that the intestines can absorb more nutrients from the food that you eat. Spheroid is another shape of a cell. Circular. These are white blood cells, and you'll find those, of course, in the bloodstream, and being circular allows for their transport in the blood more smoothly. Discoid cells, such as these ones, those are red blood cells, and they also have a large surface area. A large surface area for red blood cells, why would that be a good thing? What are red blood cells exchanging with the environment? They are exchanging oxygen, carbon dioxide. And since our cells are hungry for oxygen all the time, or they would not operate, the red blood cell has a large surface area and also trillions of hemoglobin. Stellate cells, these ones here, they have many branches, as you can notice here, lots of branches. Those are your nerve cells. their branches are functional in communicating with other cells. So your entire life, you will grow new branches, dendrites, and you will continue doing that into your very old age. Maybe you'll all live to be 100 or more, but you'll always grow new ones. And even if an individual has a stroke and some of the brain tissue dies, then you can recover from that kind of thing because the nerve cells are constantly forming new dendrites and new communication. Not the nerve cells themselves don't regenerate, but they do make more and more branching. Fusiform is another kind of cell. These are smooth muscle cells. They're also tightly packed so that they can contract in a wave-like fashion. Fibrous cells are such cells as muscle cells. So they're fibrous means they're long and thin, like fibers, and muscle cells are long and thin because they're moving the bones of your body in locomotion. So it's necessary for them to be long and thin. Well, we talked about an amoeba being a very large cell. It is a very large cell. And there are some cells that are really large. A slime mold, for example, there's a Plasmodium slime mold. Sometimes it looks like a fungus kind of. It's a very, very thin layer that sometimes you see on logs in the forest. And it can be, how big can they get? Maybe even a meter in diameter. But the reason that they can do that is that they have many, many nuclei. And they even form some membranes within the cell itself, separating the various compartments. But for the most part, cells are limited in the size that they get to. You won't find a cell that's as large as this room, for example. Most are about 10 to 15 micrometers in diameter. Egg cells are very large. Nerve cells are very long. But there's limitations. And that's due to the surface area to volume ratio. And we talked about that a little bit before. If you have a large surface area, there will be more chance for exchange of materials from inside and outside the cell. If there's a large surface area, you will lose more heat, for example. That's why you lose more heat from your hands, because relative to the volume of your hands, there's a lot of surface area. That's why desert hares, rabbits, they have really huge ears. They're flat. They have a lot of blood vessels. That's how they lose heat to the environment, that large surface area. So volume increases more. The surface area is for nutrient absorption and also waste removal. But all of the functions occur inside the cell, and the cell needs a lot of material to function. So there's a limit to how large the cell can get. If it gets too large, the surface area can't keep up with the requirements of the volume of the cell inside. Yeah, so here you've got a small cell and a larger cell. The diameter of the large cell, this is a small cell, this is a large cell, the diameter has doubled. So 10 micrometers to 20 micrometers. The surface area has gone from 600 micrometers to 2,400 micrometers. And the volume has gone from 1,000 micrometers to 8,000 micrometers. So which has grown the most is the volume. The volume increased by 8, the surface area only by 4, when the diameter increased by 2. So it's a crucial ratio, the ratio of surface area to volume. So when you look at cells, when we talk about form fits function, that's one of the forms of the cell that fits the function of different kinds of cells in the body. For example, The cuboidal cells, the square ones, they have a relatively large cytoplasm because cuboidal cells tend to be the ones that make a lot of things like hormones, for example, and histamine. So here's a general cell structure. You can see the plasma membrane. You can see the nucleus here. It's quite large. The cytoplasm is the area within the cell. You can see some mitochondria. The inside of a cell, the structures, the organelles, the vesicles, are known as the ultrastructure. So organelles, the cytoskeleton that holds it all together. And in that video that the very first video that we saw showed the cytoskeleton. A lot of those fibrous structures were cytoskeleton, and the cytosol is the liquid portion inside the cell. The plasma membrane is the structure on the surface of the cell. The plasma membrane. Hang on. I have somebody waiting here. There we go. Sorry, I have to admit someone. Okay. The plasma membrane is the surface of the cell. And its structure is so important. And it's liquid. So here is a cell up here, and this is a cell down here. This is the whole thing is a cell. This is the nucleus of the cell below. It also has a membrane around it. This is the nuclear envelope. And it is a bilayer. It's a double bilayer. So one of these here, let's see, I'll use red. One of these here is one bilayer, and this is another bilayer. That means there are two layers of phospholipids. And we'll definitely get into the plasma membrane in more detail. It's a mosaic. So the plasma membrane is fluid. It's not solid. It's a mosaic of various structures, lots of proteins. It's selective. Not everything can get in a cell. And it's semi-permeable. Some things are just simply too large to pass through the cell membrane. The proteins that are embedded in the cell membrane have many different functions. Some of them are receptors, such as this one. This one may be a receptor. And this is a ligand. A ligand is a substance that is basically floating in liquid. It can be in the blood, it can be in the interstitial fluid or the fluid between cells. The receptor is there to receive signals from the blood and from the rest of the body. So the receptor is a protein, and it's specific to certain ligands. So it may be specific, for example, to a particular hormone, like testosterone. Some proteins embedded in the cell membrane are enzymes. So this one is an enzyme. And it's working. It's a working protein, a work in progress. So in this case, it's breaking down a substance. Some proteins are channel proteins. Channel protein. This one is open. It's open for business, but this one is closed. And various processes open and close them. They may be voltage gated, which we'll find out about in the nervous system, or they may open and close due to concentration. So those are channel proteins. Some are cell identity markers, and they identify the cell largely as being your cell. So they identify your cells as yourself. Yes, the plasma membrane, I have a question. The plasma membrane is the same as the cell membrane. Thank you, that's a good question. Some proteins embedded in the cell membrane are adhesion molecules. So as you can imagine, since cells have to work together in concert quite often as tissue, for example, muscle cells, the skeletal muscle is made up of many, many muscle cells. But in order for that muscle to contract all the muscle cells at the same time, they have to be very well adhered to each other. So a receptor is a cell communicator. Chemical signals arise from the body, largely part of the endocrine system. Some of them are hormones. Some of those signals are neurotransmitters. And what's very important about receptors is that they're specific. They're called receptor, that's called receptor specificity. So they don't respond to just any signal, they respond to a specific signal. Sometimes receptor activation produces a second messenger, a chemical inside of the cell. That's called a second messenger system. So this is a table that I encourage you to look at, not a table, sorry, a figure, to look at in the textbook. This is a second messenger system. It shows the receptor, which is here, getting a signal here, but that causes this protein to attach itself to something else. It all requires ATP. It triggers cyclic AMP. That's the second messenger, and that creates a series of reactions within the cell, a second messenger system. So there are enzymes involved, kinases. They are triggered to become functional. Other enzymes then are activated, and they go through many different functions. These activated enzymes, they cause many, many different metabolic functions. They may, for example, break down glycogen in the liver. But it will be a specific function depending on the signal. The plasma membrane has pores, lots of pores. The pores are created by membrane channel proteins. Transmembrane proteins are the ones that go through the entire membrane. So for example, if this is our membrane here, this is our bilayer of phospholipids. Our transmembrane protein bisects the entire membrane. Some are constantly open. Aquapores, for example, aquapores. Those are the pores that allow water into the cell. So water, as we know, is hydrophilic. But the plasma membrane consists of lipid. So water isn't going to be invited through because lipids and water don't mix together. So the aquapores allow for water to enter the cell. They're constantly open. Some, however, are gated channels. That means they only open in response to stimuli. Some of them are ligand gated, chemicals such as hormones. Some are voltage gated, that occurs in the nervous system in particular. And some are mechanically gated with stretch and pressure. So for example, the walls of the bladder, your bladder tends to stretch, and that can cause some channels to open. Very important in nerve signal and muscle contraction. And that is because muscle contraction takes place almost instantaneously, as do nervous signals. So those gated channels have to open just like that, and they do in response to currents. Microvilli are extensions of the membrane. They're quite small. Some of them contain actin. Their function is to increase the surface area for absorption. It shows as a little brush border, and you'll see this when we do the lab. The actin kind of milks the microvilli. They shorten it. So let's see if I can draw this. Let's say this is a cell that's lining the intestines, and there is a microvillus. One is called a microvillus. That's not really very big, is it? No, hang on, I'll make that larger. Although the microvillus is small, I'm going to show it as larger, just to demonstrate. And here is a strand of actin. Actin is contractile. It's a contractile string of protein. And here's a piece of food. And the piece of food is absorbed into the cell. But how does it get from the microvillus down into the cell itself? Well, what happens is the actin shortens, and it brings that food material down into the center of the cell. Cilia are an extension of many cells. They're longer than microvilli, about 7 to 10 micrometers long. I should say micrometers. Sometimes they're called hairs. So for example, in your ears, you have hair cells, not really hairs at all. They are cilia. Some cilia are non-motile. For example, in the inner ear and the retina and nasal cavity. So in the nasal cavity, those non-motile cilia pick up odor molecules that you breathe in. In the inner ear, they are attached to a membrane that changes shape with sound. So they don't move. They're non-motile. I think they're also known as stereocilia, are non-motile. Some cilia are motile, lucky for us, and they beat in waves. They have a power stroke followed by a recovery stroke. So these are the cilia that line your respiratory tract. They line your respiratory tract. So the mucus in your respiratory tract traps particles like pollen, dust, even mite feces, which are in the air. And the cilia move that mucus out of the respiratory system that you cough up. It's a fair bit. It's like what is it now, 2.5 liters a day or something like that. It's quite a bit of mucus that we swallow, that we've coughed up, and of course from our digestive system. So the reason that cilia can pump this mucus, even though mucus is quite sticky, is because there's a saline layer at the cell surface here. And chloride pumps pump that saline into the cell surface, and it causes the mucus to detach. And that floating mucus then is pushed along by the cilia. Here's a scanning electron microscope image of cilia. This is a rabbit trachea, and you can see them here. These are all cilia. And they do this power stroke, boom, a power stroke, but then they come back in a recovery stroke. So they only beat in one direction. So a power stroke and recovery stroke. Sometimes those chloride pumps do not work. If they do not work, as in cystic fibrosis, then that saline layer does not form under the mucus. And that creates mucus plugs. Yeah, so that's if the mucus builds up in the respiratory system, then oxygen cannot make its way to the body and to the cells. So there's an inadequate absorption of oxygen and of nutrients as well. So the pancreatic ducts will also clog with mucus. So we said that the plasma membrane is selectively permeable. It controls what enters or leaves a cell. That is extremely important because cells only require a certain amount of nutrients, oxygen, but they're also transporting out of themselves waste, including carbon dioxide. So there can be such a thing, there is such a thing, as passive transport. It doesn't require energy. It doesn't require ATP. And the reason it works is that substances move from an area of high concentration of themselves to an area of low concentration of themselves. So down, in other words, their concentration gradient. So if we have, I'm going to say that's a plasma membrane. And we have lots of solutes here and very few here, then the solutes will move into this area until there's an equal concentration or it's at equilibrium. Yeah, so filtration works that way and simple diffusion. So simple diffusion works with very small molecules. They have to be very small. It also works with lipid-philic molecules or hydrophobic molecules. So in other words, steroids, lipid hormones, they can move quite readily across the plasma membrane because they themselves are a lipid. But many proteins cannot. So protein hormones, they can't just move right in. They have to attach themselves to a receptor and cause a second messenger system inside the cell. Active transport requires ATP, adenosine triphosphate, because often movement is required against the concentration gradient, particularly in the nervous system. Some are carrier-mediated, facilitated diffusion and active transport, and some transportation occurs in vesicles. So let's see what that looks like. Filtration. That's movement of particles through a membrane by hydrostatic pressure. What provides that hydrostatic pressure? Blood pressure. So your heart beats, pushes blood through your system, and in particular, some examples where this occurs is filtration of nutrients from blood capillaries into tissue. So that's as a result of your blood pressure or hydrostatic pressure. Also, your kidneys are constantly cleaning your blood, and the wastes are filtered from the blood in the kidneys due to that hydrostatic pressure. Simple diffusion is just net movement from high concentration to low concentration. Why do they move at all? Well, as we know now, atoms and molecules are in constant motion. They're constantly moving because of the wobble of the nuclei and other reasons. So movement down the concentration gradient, simple diffusion. Now, osmosis is a kind of diffusion, a diffusion of water. Osmosis is the diffusion of water. And through a membrane. So it does that through channels known as aquaporins. They're specialized for osmosis. And the thing to keep in mind that is very important is that water moves down its own concentration gradient. So where water is more concentrated, it will move into an area where it is less concentrated. Yeah, so in this case, for example, in this beaker, here's the membrane dividing, and there are less solutes on this side, fewer solutes, and more on the other side. Therefore, water will move over here because there's less water over there. So eventually it will look like this. So the water has moved in this direction. Eventually it will stop moving because the hydrostatic pressure, the pressure from, in this case, the atmosphere, prevents any more water from moving in that direction. There will still be some movement, but the net movement will be zero by that point. So we have another name for concentration. It's called tonicity. Tonicity. And it affects fluid. It affects the movement of water within a cell. So there are three different types of tonicity. One is known as hypotonic, one is hypertonic, and one is isotonic. And one thing to keep in mind that's very important is tonicity is relative. So something is relatively hypotonic to something else. So if I have, for example, a cell, I'm largely making it larger. I'm making it larger for demonstration purposes. But here's a cell in a beaker. I'll make 3 beakers. How about that? We'll call this beaker hypotonic, hyper, and iso. So if I were to draw the solutes in a hypotonic solution, there are lots of solutes in the cell, but the beaker has less solutes. It's hypo or less. So there are less solutes in the beaker. It's relative. Hypertonic, as you can imagine, means that the outside of the cell or the beaker is hyper or more tonic than the cell. And isotonic means they are equal. And there will be no net movement across the cell membrane. So these also, to remember, are non-permeable. So these solutes cannot move across the cell membrane. If they could, they would do it. But in this case, they can't. They're too large. Therefore, in this case, where will water move? Will it move out of the cell or into the cell in the case of hypotonicity? And you can write that in the chat if you like. In this case, where will water move? Into. Good, yes, it will move into the cell and the cell will start to expand. What about in the case of hypertonicity? The beaker is hypertonic relative to the cell. As you can imagine, the water will move out and the cell will shrink. In this case, a little bit will move out, a little bit will move in of water. This is H2O or water movement. And there will be no net movement in or out of the cell. So what does that mean for your red blood cells? Well, these are different solutions in which you find red blood cells. Here's hypotonic, and the blood cell is now round. It no longer has that nice discoid shape like this one does. So this is a depression in the middle. That gives it a more surface area. As we said, red blood cells require because they're constantly delivering oxygen to the body. So this is isotonic normal red blood cell shape. This is hypotonic. Water has gone into the red blood cell. And this is hypertonic, where water has come out of the cell and the cell is shrinking. Another name for that is called crenation. That is the same as shrinking. So how is that important to your blood and interstitial fluid, the cells that are in your body? Well, it's important that you don't get too hypotonic because your cells could burst. It's important not to be too hypertonic because your cells will shrink. So that's why electrolyte concentration is maintained in the body. It's maintained by mechanisms that we have of homeostasis. So proteins carry solutes across a cell membrane. That's carrier mediated. They're quite specific. A solute will bind to a specific receptor site on a carrier protein. It differs from enzymes because the solutes are unchanged. So facilitated diffusion and active transport are two of those things. So we saw them before if we have our double membrane. And here is a protein. This protein is allowing solutes to enter the cell. This may be passive, like just simply diffusion, or it may require ATP. If it requires ATP, it's known as active transport. In other words, this channel is not going to open unless ATP causes it to open. Requires adenosine triphosphate. So say you have a cell, like so, and there's lots of aquaporins, and they're open. Well, water enters the cell and fills these pores up. Now, the rate of water going into the cell is not going to increase anymore because there aren't any more aquaporins. Indeed, the proteins, the aquaporins that are there are saturated. And that's called the transport maximum. So the rate at which substances enter the cell have an upper limit. In this case, this is the upper limit. Because all of the proteins will be saturated. All the carriers are occupied. I use the term saturated, but it's the same thing. So down the concentration gradient, yeah, no problems. It's facilitated by the protein. It opens up to allow the solute to go in. On this side, this ECF stands for extracellular fluid. That means on the outside of the cell, extracellular fluid. And this is intracellular fluid, intracellular. That's on the inside of the cell. So in this case, the concentration of this solute is greater on the outside of the cell here. And so it's passive diffusion. It's going down its concentration gradient. No ATP is required here. But when this solute binds to the protein here, it changes its shape and allows the solute to continue into the cell until it's released. And then the protein will go back to its original shape. So we did talk about proteins just recently and how important it is that they have a very particular shape, and that shape can change. So this is an example of that. Active transport requires ATP. It requires energy. Some examples are the sodium-potassium pump, pumps that bring amino acids into the cell. Amino acids are enormous as far as the cell membrane is concerned. And also those that pump calcium out of the cell, because they'll be going against their concentration gradient. The sodium-potassium pump requires an enormous amount of ATP. You wouldn't believe it. Indeed, half of what you eat today, the energy coming from that will go to your sodium-potassium pumps. That's how much energy they require. They're constantly maintaining electrolyte concentration inside and outside the cell. And we'll look at that in the nervous system as well. So sodium is tiny, potassium is tiny, constantly leaking through the membrane. Half of your daily calories are used for this pump. 1 ATP is required to exchange 3 sodiums pushed out for 2 potassiums brought back in. The ultimate effect of this is that the outside of the cell will be more positive, and the inside of the cell will be more negative. So the sodium-potassium pump maintains this gradient. And it maintains the gradient so that there can be spontaneous changes in that gradient when required during a nervous signal. So ATP, of course, is a fantastic molecule that we have. Here's the ATP here. And because the adenosine is attached to 3 negatively charged phosphate groups, of course, this is very likely to break. Indeed, it will break spontaneously. That last bond will break spontaneously, producing ADP and the little inorganic phosphate. The very act of that occurring as the ATP binds to this protein causes it to change shape. That's how ATP works in the body. It's called phosphorylation. So phosphorylation is the attachment of an ATP to some molecule, causing that molecule to change shape. What does the pump do? It regulates cell volume. There are fixed anions in the cell. Anions that are large, they are negatively charged, and they don't cross the plasma membrane. But they attract cations that causes osmosis. The cell swelling due to osmosis stimulates the pump to change ion concentration and osmolarity, and cell swelling goes down. Should be, say, change. Another function of the sodium-potassium pump is heat production. So the thyroid hormone will increase the number of pumps in order to produce heat through the action of the sodium-potassium pump. Every chemical reaction in the body creates heat as a byproduct. The earth is always going from order to disorder. So every chemical reaction has heat as a byproduct. So if you have a lot of reactions, you have a lot of heat. That's why you heat up when you exercise. For example, you exercise, there are so many chemical reactions going on in your body, your muscle contractions and all that. It's crazy. It produces a lot of heat. So in the case of the thyroid hormone, heat is produced because it increases the number of potassium pumps in the body. Another function is the maintenance of membrane potential. It keeps the inside negative and the outside positive. Also, there is secondary active transport due to this difference of concentration, a steep concentration gradient. Therefore, carriers can move sodium across the cell membrane very easily. Vesicular transport is another kind of transport. It's the transport of large particles or fluid droplets. So they don't go through a transport protein. Rather, they go through by exocytosis or endocytosis. So that is when, I do have a diagram of that, but I don't think I've got a diagram of, I do, I have that coming up, so I won't draw it. But exocytosis, exo for exit, transporting out of the cell, endocytosis, transporting in to the cell. Phagocytosis is a type where very large particles are engulfed. So that is how amoebas engulf particles. That is how your amoebic white blood cells, your macrophages, engulf particles like bacteria in your cells. Pinocytosis is taking in fluid droplets. They're smaller. Receptor-mediated endocytosis takes in very specific molecules when they bond to receptors in the cell membrane. So here's phagocytosis, also known as cell eating. So here's a phagocyte and a little particle. And the phagocyte starts to build these pseudopods. They're called, pseudo means false, pods is feet. Starts to build through cytoplasmic streaming and changing its cytoskeleton somewhat, starts to surround the particle. They're called pseudopods. Once that particle is inside, it's known as a phagosome because it combines with a lysosome. And lysosomes have a lot of enzymes that will digest and break down that particle. Yeah. So it becomes it's a phagosome, it combines with a lysosome, and then it becomes a phagolysosome. And all of that particle will get broken down into inert pieces of debris that can be resorbed or excreted. And the residue gets voided from the cell by exocytosis. Receptor-mediated endocytosis occurs when molecules are required inside the cell, but they must bind to a receptor that's in the plasma membrane. Once that occurs, then the plasma membrane will involute or invaginate and eventually get pinched off, forming a clathrin-coated vesicle. Now the needed products are inside the cell in a vesicle. And exocytosis is secreting material outside. So here is a vesicle with perhaps hormones that are required outside the cell or proteins that are required outside the cell. This is a vesicle here. It's a secretory vesicle. That doesn't mean it's keeping secrets. That means it will secrete products outside the cell, and it will bind through a linking protein and then fuse, because it's a lipid. This part is a lipid. The plasma membrane is a lipid. It will easily fuse to the plasma membrane and then excrete particles outside the cell. That's exocytosis. So I'd like to end now. Thank you for watching.